Welcome to the GlycanTrigger podcast series. Over the course of 12 episodes, we will engage with experts in inflammatory bowel disease, patients, and other relevant specialists, delving into their work and exploring the challenges within the GlycanTrigger project.
Welcome back to the GlycanTrigger podcast series. I am Harry Sokol. I am professor of gastroenterology in Antoine Hospital, Sorbonne University in Paris, France. And I'm involved in the GlycanTrigger project, mostly regarding my expertise in gut microbiome in IBD. So if you are listening this podcast for the first time, GlycanTrigger is a project funded by the European Union through the Horizon Europe Research and Innovation Actions. The project aims to unravel the complex network of factors that contribute to the transition from a healthy state to a chronic inflammation in the gut. And in this series, we bring you biannual discussions featuring experts in IBD, as we explore the latest research, treatment strategies, and lifestyle factors influencing gut health. So in today's episode, we'll be exploring the concept of diet as therapy. We'll also gain insight into from the My Gut Health project, which is another EU-funded project under the same topic called as GlycanTrigger. So the My Gut project is led by Institute of Clinical Molecular Biology, University Hospital Schleswig-Holstein, and it aims to develop a personalized blueprint of intestinal health to predict and prevent IBD. The overall goal is to discover how diet, lifestyle, and microbiome-related intervention can be used to personalize treatment and improve outcome for IBD patients. To guide us through this discussion, we have two great invited speakers. So Dr. Sina Hübener, who has been the director of the Department of Medicine at the University Medical Center Hamburg-Eppendorf since 2019. He also heads the section of molecular gastroenterology and immunology. He holds board certification in Internal Medicine and Gastroenterology, Infectiology, Immunology, and Transplantation Medicine, and his research focuses in immunoregulation, IBD, and cancer. And the second invited speaker is Dr. Sina Hübener. She's a medical doctor and resident in Internal Medicine and Gastroenterology at the University Medical Center Hamburg-Eppendorf. Her clinical and research work centers on autoimmune liver disease, celiac disease, and gluten sensitivity, with contributions to clinical trials and publication in leading medical journals. So welcome to both of you, and thank you for joining us today. To start the discussion, I just want to say that obviously there is growing evidence that diet plays a critical role in the development in IBD and also in the management of inflammatory bowel disease. The food we eat not only provide nutrients, but it also shapes the gut microbiome. It has an impact on our immune function and can even modulate inflammation directly. Obviously, physicians, but also patients are increasingly interested into how specific dietary pattern can play a role in the treatment, in the care of the patient, notably a Mediterranean diet. And it can also be used potentially as a therapeutic tool by itself. So at the same time, we have several projects such as GlycanTrigger and My Gut Health that are advancing our understanding on how the difference in microbiome and immune response can guide personalized nutrition strategy. So with that in mind, we can explore together how diet can interact with the gut microbiome and what can be the consequences for the care of our patients. So maybe, we can try to discuss several topics. Maybe the first topic can be the connection between diet and the gut microbiome. So here, Sina and Samuel, I invite you to tell us what you think, notably about the influence of the gut microbiome in patients with IBD, and also if you think there are specific dietary patterns that are promising to modulate inflammation in IBD.
Okay, thank you first of all for the warm introduction and welcome. I think we are discussing a very important topic today. I mean, we have done in the last decade major breakthroughs in terms of basic research, deciphering the connection between the diet and the microbiota. So we have learned that the diet is one major factor modulating the intestinal microbiota, and actually the changes in dietary patterns, they translate very fast to changes in the intestinal microbiota. That means within days. But it's not only important what we eat, but it's also important when we eat. So we also understood that the time point when we are eating is the time point when we are feeding also our microbes. And this feeding rhythm also induces a circadian rhythm in our intestinal microbiota. So it's not only important what we eat, but also when we eat. And in terms of what we eat, I think we have learned several things. We have learned that, for example, a diet which is a Western type diet, a high caloric diet, in principle what it does, it impacts the microbiota, and it impacts then the immune system in a way that our intestine gets more leaky. So this high caloric diet basically opens the tight junctions in the intestine, which from an evolutionary viewpoint made sense because it allows us to absorb more nutrients. But of course, if you now have people who are susceptible, for example, for IBD or who are currently in infection, then of course you may actually induce something which is not good for the patient. You may actually promote inflammation. And on the contrary, we found that a diet which is rich in fiber basically strengthens the intestinal barrier and has a beneficial effect on the immune system. But this is all for basic research and basic research questions. I mean, if we now think what are promising dietary patterns, for example, in patients with IBD, then it becomes more complicated. And that's also part of the reason why we are doing the MIGAT study, to decipher specifically the impact of dietary gluten on the outcome of IBD. Maybe, Sina, you want to say a little bit more about the dietary patterns and recommendations in general for IBD patients.
Sure. I want to thank for the warm introduction as well, and say hello to everybody. So numerous studies have shown that dietary patterns indeed have significant effects on the development of IBD. And growing body of evidence suggests that the effect of the dietary pattern on IBD may be attributable to the alteration of the gut microbiota and gut microbiota-derived metabolites, because the gut microbiota acts as an effector between the diet, the IBD, and bacteriophagity maybe serves as an antigen to modulate the host immune response. So this is quite interesting, and it seems that that some of the dietary patterns indeed worsen diets and can lead to dysbiosis. So some pro-inflammatory diets, such as the Western diet, the high glucose diet, and high fat diet, high salt diet, consistently reduce the abundance of beneficial species and promote dysbiosis, and then can activate intestinal inflammation, immune imbalance, and eventually also exacerbate colitis. And on the other hand, there are plant-based dietary patterns, such as Mediterranean diet and vegetarian diets, that can indeed reduce the abundance of pathogenic bacteria and consistently increase the abundance of beneficial species, and thereby increasing the concentrations, for example, of short-chain fatty acids. And specifically in IBD patients, we found in recent studies that a significant numbers of IBD patients from the clinical part report an improvement on gastrointestinal symptoms under gluten-free diet, despite not suffering from celiac disease or non-celiac gluten sensitivity. And only a small number of studies are available, and most data come from retrospective studies. Our previous basic research studies that we could indeed demonstrate that gluten aggravates DSS colitis, and a long-term gluten-free diet could protect mice from gluten-aggravated colitis. And more importantly, in germ-free setting, it seems that this effect is also mediated through microbiota alterations after the long-term gluten-free diet intervention. So therefore, in the MIGAT project, which is an investigator-initiated prospective clinical study with one interventional arm in IBD patients, which is performed at the UKE. We perform a long-term dietary gluten-reduced normocaloric balanced diet, which is advised for 78 weeks to patients with ulcerative colitis and Crohn's disease and PSC-IBD patients. And patients are trained by clinical dietitians. And yes, we are currently recruiting at the end of the phase for the study, and we will be very, very interested in the microbiota development and the effect on the microbiota and on the intestinal inflammation in these patients.
Okay, yeah, that was a very good summary. Should we go to the second topic, immune system and nutrition and IBD?
Yeah, absolutely. We can move to the second topic about immune system and nutrition in IBD. So the two questions are to know whether diet affects the interplay between the immune system and the gut, and if you think certain nutrients or food group may be interesting to directly support immune regulation in patients with IBD.
Mm-hmm. Good. So I think in general, we have cells which are inflammatory and cells we have anti-inflammatory in a very simplified way. But then there comes a third aspect, which is particularly important in IBD, which is the impact of the immune cells on the barrier function. So we have some cells which produce cytokines which strengthen the barrier function, and then we have other cells which basically weaken the barrier function. So I think these three aspects, in pro-inflammatory, anti-inflammatory, and effect on the barrier, they have to be taken into consideration. And now when it comes to the diet, then of course the diet on one hand provides antigens, which can modulate the T-cell response. So dietary antigens, for example, also gluten could potentially do this. And then we have indirect effects, which are made via the intestinal microbiota. And also in terms of the intestinal microbiota, we have two effects. On one hand, the microbiota also serves as an antigen for the immune system, which is recognized by the T cells. And on the other hand, the microbiota produces metabolites, which then shape the immune response. And what we have learned in the past is that the antigens play a key role during T cell differentiation. So T cells are one subset of the immune system which plays a particularly important role in inflammatory bowel disease. And these antigens, they play an important role during T cell differentiation in these different subsets, which are then either inflammatory, anti-inflammatory, or impact the barrier function. However, it's not only T cell differentiation, but we also learned that the T cells have the capacity to change their phenotype. This is a phenomenon which we call T cell plasticity, or the functional adaptation of the T cells. And it seems that this functional adaptation or plasticity of T cells is then modulated by the metabolites, which are provided by the intestinal bacteria. So that means if I change now my diet, I have direct and indirect effects how the immune system is shaped. In general, one can say if you have this diet which is basically not as good for the intestine and may even promote inflammation, it has the aspect that it promotes the inflammatory T cells and at the same time weakens the T cells, which are anti-inflammatory, and it also weakens the immune system, which is important to tighten up the barrier function. And this is, again, these are diets which are basically high fat diet, high caloric diets, versus the diets which are more rich in fiber. They promote the anti-inflammatory T cells, and they also promote the part of the immune system which is important to tighten up the barrier function. So this is the general or basic research, what we have learned in recent years. And maybe I hand over to Sina for the nutrition for IBD patients, what we can do. Sina, do you want to catch up here and now say how we can translate that into humans? Of course, for humans, we know much less mechanistic details compared to the mice, because also there's a lot of things that we can do in experimental system, which we cannot do in humans. But still, due to the single cell sequencing techniques that one has established in the last years, we have also gained more knowledge in humans.
Yes, sure. I can give an update on what is already known or what is rather being recommended for known dietary recommendations. So although they, as Samuel already said, the exact mechanism by which dietary intervention affected the IBD remains unclear, some plausible explanations have been proposed. And to sum this up is that the diet may modulate the mucosal barrier function, that the diet may be involved in the inflammation response and immune function. And the third part is the diet may contribute to shaping the gut microbiota and its metabolites. And what is known for diet or which dietary recommendations are out there right now are, for example, the Western diet, as we already mentioned before. So the Western diet, in principle, increases the permeability of the epithelial barrier by inhibiting the or causing intestinal so the dysfunction and the excessive release of pro-inflammatory cytokines seems to be lowered in this diet due to the less intake of sugar and the higher intake of vegetables and fruits, for example. And other vegetable dominant diets are, or rather say plant-based diets who contain fiber and especially the extra virgin olive oil. So these IBD diets should be recommended in IBD patients. In addition, I can say more about the specific carbohydrate diet. So this in this diet, this permits the consumption of only monosaccharides in patients with IBD based on the theory that disaccharides and polysaccharides enter the colon without digestions, resulting in excessive growth or bacteria and fungi and secondary harmful exhales. And in this diet also grains and processed meats are excluded. So one problem in these diets is the poor adherence from side of the patients, and that patients can develop some nutritional deficiency. One more diet which is sometimes recommended in patients with IBD is the so-called low FODMAP diet. So the low fermentable oligosaccharide, disaccharide, and monosaccharides and polyol diet. The theory behind this is to eliminate short chain carbohydrates that can be fermented by the gut microbiota, which results in gas and bloating and abdominal pain. and changes in bowel habits. And so this can improve the clinical symptoms in IBD patients a lot, but this also should be only performed under the supervision of a dietitian, because there's a risk of micronutrient deficiencies and also on malnutrition. And last but not least, there's a special diet for Crohn's disease patients, so-called the Crohn's disease exclusion diet. And especially in children, there's the enteral or exclusive enteral nutrition, which is only a fluid diet that these patients are only on the exclusive enteral nutrition. And the Crohn's disease exclusion diet is a whole food diet that includes fruits, vegetables, and meats, and both simple and complex carbohydrates, but eliminates certain animal fats, certain types of meats, and reduces exposure to food additives, for example. And this seems quite helpful in some patients. So to sum this up, there's another option, as we already told you about the gluten-free diet, which eliminates the gluten and which exists mainly of wheat, barley, rye, and triticale, and related processed foods. Because, as I already told you, some of the patients with IBD are gluten sensitive, and even in those which are not so-called gluten sensitive, a number of patients report about the improvement of gastrointestinal symptoms, despite not suffering from celiac disease or non-gluten sensitivity. But the adherence to this diet is also difficult. And so I think this was a lot about the available dietary recommendations.
Okay,
Just
as a comment, I mean, for now, gluten-free diet or low FODMAP diet are not recommended for patients with IBD. This is very important to be clear that this is only for research purpose for now, I guess. So just to connect with shortly with the next topic, because I think you covered already a lot. So in clinical practice, is there some dietary recommendations that we can already give to the patients, or from your point of view, this is only a matter of research?
Yes, so the definite recommendation out now is the Mediterranean diet, or rather the plant-based diet, which contains mainly fiber and the extra virgin olive oil. But there's a limitation, especially in those patients who are during flares. This diet has definitely problems with digestion. So easily digestible food, low in fat and low in fiber, is recommended in patients with a very active form or active phase in those patients. And so with moderate to high inflammation. But the definite recommendation for patients who are currently in remission or almost remission would be the Mediterranean or plant-based diet for now.
I fully agree. Just want to highlight that there is consensus that is on its way and should be published hopefully this year on really all these aspects about dietary recommendation in IBD, and this is fully in line with what you just said. Maybe to move to the next topic, we talk of course a lot about diet. This is clearly the topic of the day. But is there other environmental factors that are involved in IBD?
Yes, so maybe I continue. So what we have learned in the last years is that the so-called exposome, that means that is everything which from the outside impacts our body, plays an important role, and diet is of course only one component. So there are other factors which play a role, and this is lifestyle. That means not only what I eat, but also what I work. For example, if I have a regular work or if I have to work in shifts, that can impact, of course, my health. And also the environment that can impact us is, for example, the pollutants, but also the microplastic. So there's some basic research data which is now deciphering the exact impact of pollutants or microplastic on the immune system and then on the susceptibility to develop IBD. But also, of course, we have the observation that the increased incidence that we have experienced in the westernized country, in the industrialized countries, is now also happening in the newly industrialized countries. And that suggests that it's not only the diet or the genes, but it's also the whole environment which can impact our immune system and therefore the susceptibility to inflammatory bowel disease. And as I said, the two aspects which might be important are the microplastic and the pollutants. At least the basic research is pointing into this direction. But of course, we need more translational studies to really assess the impact on the human disease.
Thanks a lot, Samuel. So maybe we can move to the last topic of the day, which is basically the future. How are we going to take all this science, this knowledge about diet, effect on immune system and on IBD in the future? How are we going to basically incorporate these type of approaches in the current, with the current drug we are already using, and what are the gaps basically to be more practical and to really use the diet in our care of patients with IBD?
Yes, so what we want, I think, are two aspects. First of all, there are several studies which assess the impact of specific dietary patterns, like we do it for gluten. And that, I think, is certainly one important aspect. But of course, this is still based on the whole population of IBD patients. What we would like to have in future is a more personalized dietary recommendation. There are approaches, not in IBD, but in for example, metabolic syndrome, where thanks to big cohorts and machine learning algorithm, one is already able to predict, based on the intestinal microbiota, the ideal dietary pattern to have a low blood glucose level after ingestion of food. Of course, the advantage in that system is that the blood glucose levels are very simple to measure, and that is why it was more easy to basically have personalized dietary approaches which are aimed to lower the blood glucose level. In IBD, it's more complicated because what we want to do is, of course, to lower the intestinal inflammation. And the intestinal inflammation cannot so easily be measured like the postprandial glucose levels. And that's why it's much more difficult to establish a personalized dietary approach in IBD patients. But of course, dietary approaches are only one aspect to modulate the intestinal microbiota. There are many other aspects which are now tested, and one would be the phage therapy. Phages are able to specifically kill specific bacteria, and we are now getting better and better to learn the role of specific pathobionts. So pathobionts are bacteria which belong to the normal commensal microbiota, and they don't make iron sick per se, but they increase the susceptibility to certain diseases. So there are certain pathobionts which are okay in healthy individuals, but they may increase the susceptibility to IBD, or they may increase the IBD severity. And now, of course, we are getting better and better in identifying these specific pathobionts. And then the idea would be to specifically eliminate these pathobionts, for example, using phage therapy. So this is one possibility how we maybe in future able to alter the intestinal microbiota in a more personalized way. Either we have personalized dietary recommendations based on the intestinal microbiota, or due to personalized interventions, for example, via phage therapy to modulate the intestinal microbiota. But at the moment, we don't have either of those for the IBD patients.
Thank you very much, Samuel and Sina. Thank you very much for all your comments, your explanation. I think it was great. Hopefully, both the My Gut Health project and the GlycanTrigger project will help us, will learn, will teach us some important lessons about how the microbiome interacts with diet, with the immune system, notably for the My Gut Health project, but also with the glycan structure for the GlycanTrigger project. There is obviously some complementarity between both of the project, and obviously it's critical to be able to collaborate to get most of our results. And I'm very happy that both the two consortium are really collaborating and communicating. Do you have anything you want to add?
No, I think you have mentioned the key aspects. I think there's much to learn in the field of IBD. We are getting much better in terms of basic research. But now the challenge of the next year will really be to translate this basic research into clinical applications. And I think the interaction of the different consortia will be critical to achieve this, because we all basically try to understand the impact of one specific dietary one specific part of our dietary. So only if we take all this knowledge together, we will be able to help our patients. So that's it from my side, and thanks again for having us here.
Thank you very much to both of you, and thank you to everyone who is going to listen to this podcast. I hope you enjoyed it as much as us, and so bye-bye everyone.